or you can get too overwhelmed. There's a lot of stuff that you have to go and break down each individual thing. So this is just an intro broad overview. So roles of clinical virology. Why do we do virology? If you still do virology, a lot of places they cut it out, but it's still there in the public health, and I think active children still may have it. I'm not sure.
Yeah, they do.
So what we do, we provide diagnosis. So we need to distinguish between an active and a latent infection. Viruses can lay dormant or latent in your system for a long time, if not the entire lifetime. So we need to know if this is something that's currently causing a problem or if this is something that is just kind of hanging out. We also need to know what the nature of the infection is. While this can count, sorry. We need to know what's causing the infection. So this is more specifically, I mean, not more specifically, but this pertains more to respiratory cultures, where you have a far greater number of viral pathogens for respiratory than you do, let's say, a leg wound or arm wound. Also with like urogenital tract infections and neonates, those also will have a lot of viral implications as well as bacterial and fungus. So we need to be able to differentiate between those. How to treat, obviously, a antibiotic is not going to work for a virus. So we need to make sure that the treatment's going to work and what kind of treatment we need. We don't test per se antivirals like we would antimicrobials such as like penicillin and stuff like that for bacteria or azoles for funguses, but they're pretty type specific. So, retroviruses get one type of drug, they get antiretrovirals, and then, the herpesviruses have a different type of viral. So we need to know what it is so we can treat with the right thing. So identifying the virus is important. Was something flashing or am I just looking?
Oh, it's flashing. It's like blinking on the screen. It's like, yeah.
Like you're having these nuts.
Jen, is there a way that the cahoots it can like have us access like the quiz one?
Oh, what's it taking you to?
It takes us just to the screen where it's like the like
Oh, it's taking you to like my screen. Hopefully I showed you the right thing.
I think you did.
Yeah, you did like the start button that plays it.
Stop. I didn't know it's still doing it.
Yeah, doing it.
Let me fix that.
Something we do for the face.
All right, well, let's just keep trucking because I'm not a PC nerd, microbiologist. So, anywho, surveillance, public health, bioterrorism, stuff like that. During the COVID pandemic, we had a lot of this going on. Every year during flu season, I would have to count up our positive flus and send it to the Summit County Health Department, and they would put it in their statistics. So for some reason, there's a whole bunch of people that love to do that. More the power to them. I guess they're just trying to prevent outbreaks, which is a good thing. Patient progression, yeah, viruses are kind of hard, especially because nowadays we use PCR to do most of our detection. And the problem with PCR is it doesn't matter if you're active or latent, it's going to be positive. So really the only true way to do patient progression is viral cultures, but a lot of people don't do that. So it's a double-edged sword, PCR. So viruses versus bacteria. If you haven't gotten this already, I have failed you in bacteriology. Viruses are a lot smaller, where in bacteria we get to see on plates and under the microscope. they are alive and you treat them with antibiotics. Viruses are submicroscopic, so we don't see them. We don't see them specifically on microscopy. We are actually looking for the cytotoxic effect that happens when a virus infects a cell. So that's what we're looking for when we look at microscopy. An electron microscopy, different ballgame. This is talking light microscopy. They lack cell walls, metabolically inactive. Basically, we talked about they can't be treated with antibiotics because they're not alive. So the definition of a virus are small submicroscopic obligate intracellular parasites. Basically, they need a host to live. They can contain either DNA or RNA as a genome, and they can be protected by capsids or encoded protein codes. They cannot carry out their life by themselves without a host cell, so they have to be inside a cell to live. Well, to replicate, I should say. They don't really live, which is confusing because it's not life sustaining if it's not life, but they need a host. They're like any other parasite. They need something to help replicate the cycle. They can't synthesize their own proteins because they don't have ribosomes. So they use the host cell ribosomes for translating their mRNA, mitochondrial RNA into proteins or messenger RNA. mRNA. Viruses can't generate or store ATP either, so they use the metabolic functions of the host cells. So just a couple fun facts about viruses. There's 10 to 31 to 10 to 32 viruses in the Earth's atmosphere. That's a big number. I don't really want to write it out. But there's a lot. Most of them are phages, non-pathogenic to humans. Some really cool stuff on phage therapy, if anybody's really into viruses. Really interesting stuff. We're using viruses called bacteriophages to actually treat multi-drug resistant organisms in patients where all other antibiotics have failed. So if you're into that weird nerdy science stuff about micro, look up phage therapy. Look up, there's a book, I forget what it's called. I always tell people about it. It's a really good book and I'll figure it out at the end if we have time. So basically, if you line all those up, it's about 200 million light years into space. It's pretty far. But out of that many estimated, there have been 4,000 viruses described, classified into 71 families, which we will not cover all of those because they don't pertain to us. First influenza virus isolated in 1933. In 2005, the 1918 pandemic influenza virus was constructed from nucleic acid sequencings obtained from the victims of that disease. I'm pretty sure it is illegal to complete that genome and recreate the 1918 influenza strain. Pretty sure it's a class A bioterrorism pathogen. So, parts of the virus. We have pore, capsid, and a capsimer. So your pore is what it's made of. It's either going to be a DNA or RNA virus, and it can be configured into various different forms. Your capsid is your protein. It's the protein that encompasses the nucleic acid, and then the capsimer are your individual building blocks. So you can see down here, get a little pen out, a little laser pointer. Your capsid. You take one capsid, put it together, you make a capsimer, and then you put that all together, get a full. Oh, okay, so this is just the protein. Take the protein to put it together. Okay, that makes sense. I looked at it, it's like. So a nucleocapsid is the total structure of the genome within the capsid. So this is on the inside around the nucleus. must have nucleic acid for this term to apply. And then we have some viruses that are called enveloped viruses. This is a external lipid membrane which surrounds the entire capsid. These are derived from the host cells. So as it goes in and it seals the lipids from the cells, and then it's released into the. Oh, this is an example of a herpes virus, Viron. One thing that I was very confused about when I started learning about viruses is I thought herpes viruses were just herpes, but it's not. There's a whole group that are called herpes viruses that you don't consider herpes, but they are. So don't get set in the fact that herpes viruses is like HSV1 and HSV2. Epstein-Barr and all those other ones are classified as herpes viruses. So don't make that mistake that I did, because that really confused the heck out of me. So we can see here we have our proteins on the outside, our envelope, genome in the center, and then the capsid. This thing that was just on the picture. All right, so how we classify viruses. This is also super confusing, and there's a lot of different things, and it took me a while to figure out. So I tried to make this PowerPoint a little bit easier, try to help explain what it means, because I know I was super confused when I went through this. So obviously we have some sort of genomic aspect. So it's either a DNA virus or an RNA virus. Then you can have double-stranded or single-stranded. And then there's a sense of single-stranded, so it's either a positive or a negative. So we'll get a little more in depth here coming up soon. But just know we have the DNA or RNA viruses. It's either single-stranded or double-stranded. And if it's single-stranded, it's going to be positive sense, negative sense. You have a capsid morphology, so if it's hedral, helical, or complex, we get some more examples of those coming up here soon. And then the envelope, the outer coating, so it either is enveloped or non-enveloped. So your the virus core for your DNA viruses, you can have single-stranded DNA, or you can have double-stranded DNA. So you'll see these denoted with viruses as SS, single-stranded, and then DNA, DS, double-stranded. And then for the RNA, we have the positive or negative sense, so there'll be a little plus or minus in front of it. So you can have positive, negative, single-stranded RNA, or you can have double-stranded as well. We don't. Okay, so what does all that nonsense mean? So senses, the sense, positive sense, the RNA present in the genome will be the same polarity. So I don't know if y'all have to take biochemistry and all that, but knowing like polarity, and I guess that would be organic too. Knowing all that. So positive sense just means that the RNA present will be the same polarity as the mRNA that it will encode. RNA polymerase can be immediately translated into viral proteins by the host cell. So basically, if it's a positive sense virus, it doesn't have to do anything. It can just, get into that cell and start pumping out product. The negative sense, the genome acts as a complementary mRNA strand. So you have to start that whole transcription process over before you can translate those proteins. So you can't convert the genome into mRNA without an active viral polymerase. So if it's positive sense, it's already ready to go, primed and ready to go. If it's a negative sense, it needs a little bit more work. So kind of think of it as it's negative, it's, behind the 8 ball a little bit. You need a little bit more work. All right, so we have the different shapes. Our helical are polyhedral, spherical, and complex. Most of our respiratory viruses will be spherical. Most of our helical ones, those tend to be plant-based viruses. And then polyhedral, your adenovirus, those are some respiratory and GI, depending on which one you're looking at. And then your complex, this one right here, this is the one I was poorly drawing for fun. That's your bacteriophage. So those are viruses that attack bacteria specifically. So ischihedral, they're capsids that have 20 faces, named after the 20-sided shape ischihedra. Real clever. The filamentous capsids are named because they're linear, thin, thread-like. They can also be called rod-shaped or helical. And then your head tail, that's one of the complex ones it's like a hybrid between the filamentous and the ischihedral. Basically, they're the two put together. So, real good so far? Any questions so far? Shapes, capsules, what's inside the virus? All right, now we're going to talk about how they spread. So hopefully, everybody here has a little bit of common sense when it comes to how some of these things spread. Anybody know what time of the year it is right now? It's flu season. Microbiology's favorite time of year. Second to pandemics. Pandemics suck really bad, as most of us found out. So our number one route of transmission of viruses is respiratory. Big shocker there, I know, everybody. Droplets, aerosols, secretions on the hand. Everybody's sneezing, everybody's coughing, everybody's touching things. If you have a toddler like I do, they like to go to school and they got a bunch of little kids that are coughing and touching things together because they don't understand microbiology, no matter how much I try to instill it in her. She comes home and what's the first thing she wants to do? Daddy. So guess who gets sick all the time? Guess whose fault it is? Respiratory transmitted respiratory viruses. That's whose fault it is. Or my fault for him. Anywho, so you all are pretty confident that you know how respiratory viruses transfer. I don't have to explain any more than this. Okay, good. So influenza viruses, BZV, rhinoviruses, adenovirus, RSV, paraflu, metapneumo, coronaviruses. God, I don't even want to say that word. Fecal oral viruses. These are a little bit more nasty ones. I don't know if anybody's ever seen some of those videos of rotavirus, and norovirus, and astrovirus, where the patients are just laying there. It's gross. Or if you've ever had a child, infant rotavirus, pretty big. But one that people get a lot of and hear a lot of is hepatitis A. Anybody Chi-Chi's fan? I don't know. You guys might be too young for that. But hepatitis is spread through eating contaminated vegetables and stuff like that. So those are some of the common ones. There's obviously a lot more. And then you have your direct contact viruses, your HPV, your HSV1, so on and so forth. Sexually transmitted viruses, your HIV, your HLTV1, Hep B, HPV 16 and 18, and HSV2. Hopefully you're all aware there's an HSV1 and 2. One's like the cold sores, 2. We'll cover that a little more. Urine-associated transmission, that's your CMV. We get this a lot from, well, we don't get it a lot. We have to send it over there now because I don't work there anymore, but over at kids. It's very common in kids. We have transmission through blood products, tattooing, scarification. Typically your HIV, your Hep B. That's why I always go to a reputable tattooer. I've seen some pretty nasty tattoos that have gone rogue from poor sanitation or reusing of needles. Sorry, but I paid way too much money for a tattoo to have them use a used needle. And then congenital germline, so it's from your mother to baby. Fortunately, HIV is how a lot of these babies get it. Now it is quite preventable early on, if I do believe. But the problem is a lot of the time the parents don't know they have it. Or they just don't care because they don't know they're pregnant or they don't want to be pregnant. It's really sad. But anyway, you all can kind of get what's going there. Arthropod-borne transmissions. These are the nasty ones. So anytime you have an arthropod involved, it's usually not good. So arthropods, your mosquitoes, ticks, sandflies, these are going to be your fever viruses and encephalitis, Japanese encephalitis, West Nile virus, dengue fever, yellow fever. I mean, it goes on and on. If you heard of a pandemic somewhere or an epidemic or some sort of plague, probably came from an arthropod. Zoonotic infections, rats, that's your Lassa fever, your hantaviruses. Bats, I don't know, bats have gotten a lot of attention. Bats carry rabies, Lassa viruses, Ebola, and our favorite SARS coronaviruses. Who? And then others, you have cows and sheep and monkeys. Monkeypox, that kind of hit the news recently. Cowpox. All right, we good up to this point? Pretty good. Everybody kind of semi-understanding what I'm talking about? Cool. Viral replication. So this is where a lot of the important stuff comes in. Knowing how viruses work, and then if we know how they replicate, then we can figure out how to stop them from replicating. We need to know where and when things happen, so we know what to do to go in and stop it, put like a little stopper, and before it can start the next process. So there are 7 stages of viral replication. We have the initial attachment, penetration, the encoding, replication, assembly, maturation, and then the release. So obviously your attachment, you're going to attach to whatever you're looking for. You're going to penetrate through the wall. You're going to open up. You're going to replicate your pieces. Then you're going to put them all together. Then you're going to grow them up, and then you're going to let them out in the world. Anybody Star Wars fans? Like the cloning? Yeah, there we go. Somebody in the back. You know, they go in, they replicate all the clones, then they put all the gear on, and then they teach them and then cure them to get real big, and then they send them out. Destroy empires. Which turns out to go against them. But anyway. Attachment. So the viruses will enter the cell by attaching to one of the cell receptors. I don't know how many people have taken clinical immunology. I know YSU does. Any YSU people in here? Beautiful. So you've all had Dr. Fagan. She's still there? Who teaches immunology now?
Dr. She's new.
Yeah. I love
She
Dr.
teaches
Fagan.
everything.
Dr. Fagan was a med-tech before she went back to school. She was awesome. Anywho, so basically you're going to have these viruses just kind of floating and blah, 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 blah down the surface. And eventually it'll come to a cell receptor. And the viruses, certain viruses have different, certain cell receptors. So everybody that hasn't really had immunology, your body has, it presents proteins, and then they're like locking keys. So one, it has to fit perfectly to connect. And then once it hits that receptor, it like triggers it to get pulled into the cell. This is why some viruses are more adaptable to affecting humans. For example, SARS-CoV-2. For some reason, it was, had a great affinity for the lining cells in the lung, the, what was it, HLA2 receptors in the lung. So it was a perfect lock and key match. So if you were to inhale that virus would be exposed to this surface, basically, and then it would find that specific receptor and bind to it, and you could not get it off. And that's what made COVID pretty nasty for a lot of people. Now, me, I was exposed to it and never got it, so I don't know. Maybe I don't have the receptors. Anywho, there's some different receptors that they can attach to. So next step, penetration. This is just crossing the plasma membrane. We can do this from a couple different ways.
Sorry, I lost my.
Oh, dare you agree? So anywho, before we were rude here. Just kidding. We have 3 different ways we can penetrate into the virus. We can do it through endocytosis, phagocytosis, or fusion. So endocytosis, receptor-mediated endocytosis occurs when the receptors on the cell are bound. So as I said, as they kind of roll across, they bind. It sends a little signal to say, "Hey, we got a match. Let's pull it in," and then it gets brought into the cell and put in a little vesicle. Phagocytosis, phagocytosis, I hear both ways. Say it however you want. This is a receptor-mediated endocytosis that used by specialized cells to engulf the entire cell at one time. So it just kind of goes, eats it. And then fusion. So fusion is just when the envelope of the virus feeds to the cell wall and they fuse together, and eventually it just kind of melts in or forms into. Well, here I have a picture. How about that? Pictures are always better. So we have fusion. The virus just kind of lands there, and eventually it will go and fuse together. We have our endocytoses right here, where it goes into. So see how we're going into the cell, into the cell. Endo means inside, into the cell. You can do this bulk phase endocytosis, but we're going to pretend that was not there. And then the phagocytosis is when the cell actually goes out and grabs it itself. So endocytosis, it goes in from the surface. Phagocytosis, it kind of goes out and grabs it. So uncoding, this is just a breakdown of the capsule or the capsid. This causes the genome to be released into the cell. Replication, the process of replication is dependent on what type of virus it is. So you have a positive sense single-stranded RNA virus. The infectious RNA produces mRNA that is directly translated. So it starts off and goes right away. And then your negative sense single-stranded and your DNA viruses are produced by transcription, then translated into the protein. So there's that extra step. I also believe this is why, respiratory viruses, things that are positive sense single-stranded RNA viruses, that's why they can infect people so quickly and they can spread so quickly, because they don't have to do that extra step. Or things like your DNA viruses, like your HIV and stuff that are slow over time viruses, they just take so long. Replication, synthesis of the components. These will occur inside the host organelles. So translation of mRNA into the virus protein, it uses the host cell ribosomes, the tRNA and the enzymes that are present. Replication of the genome occurs as soon as the virus polymerase has been translated from viral mRNA. then the late mRNA structural proteins, so they're synthesized after viral DNA replication has commenced. Coding for structural proteins transported to the nucleus cell membrane. A lot of nonsense. So basically, it just has to follow the typical genetic building code. Doesn't really have anything different other than it can either start right away or it needs to be translated first. And then modifications of protein, the pre-assembled capsomeres can then join to form procapsids, empty capsids, and then can be used for shell. So assembly, you have the term virons. Virons are the complete virus particles that are produced by the assembly of the preformed viral components. Main function is to deliver its genome to the host cell. So they're like little drone bees, really. You know, you have your queen bee and makes babies and sends those babies out to get what it needs, and then the cycle continues. So assembly can take place in a couple different places inside the cell nucleus, the cytoplasm, or the plasma membrane. Self-assembly is when they spontaneously assemble. And then a viron is officially completed when the nucleic acid is completely enclosed in the capsid. Maturation, all the final changes within the immature virons have resulted in an infectious viral particle. The structural capsid changes are often involved and can be mediated by host enzymes, virus activity. Basically, they just grow up. And then once they grow up, it's time for them to leave the nest. Unfortunately, when they leave the nest, this is where they cause the most damage. Final step, obviously, of the replication cycle is to release them out and start over again. So the viron particles are released into the extracellular environment. So how do we do this? We can do budding, we can do exocytosis, and we can do the always fun cell lysis. So this is just kind of the diagram of the maturation of an HIV cell. And this is the, oh wow, I think slides, hold on, we gotta go back and talk about these. I thought it would tell us, but it doesn't. So, budding, we can see down here in this picture real quick. Basically the virus just go to the surface and they sort of bud out. And then it's kind of be released into the environment. Very similar to exocytosis, the way it was brought in. You know, comes to the surface, it gets picked up, and then it goes out, or it comes in. Same way going out. It's going to hit a receptor protein, and it's going to be flagged as something to leave the cell, and then it's going to be released naturally by the host cell. Last is the cell lysis. These are the ones that are the worst because cell lysis means cell death. So instead of being nice and being patient, letting the cell release it slowly into the environment without killing it, they're like, screw this, or hop and ship, and they bust out of the cell. This is what causes death of the cell, the inflammation and stuff that you get with a lot of viruses. And this is how viruses cause a lot of damage. You know, you get viruses in important tissues like, you know, heart muscle and stuff, and you have cell lysis, but your heart muscles are dying. Oh boy, you know. You start having lung damage, and you can't breathe properly, you start losing oxygen. So cell lysis is obviously probably the worst of this stage, but I think obviously have a bunch of different phases to it. This is just showing the HIV budding out. Why HIV can lay dormant, because HIV buds, it doesn't cause that immune reaction, that inflammation of the cells slicing and bursting, so you don't really know you have it. It just kind of silently replicates until it's too late. Here we go with the life cycle of the influenza A virus. So we can see here that the virus comes in, it's endocytosed, breaks down, gets in there, replicates, does all that fun stuff. Does all that fun replication, comes out, and then it gets budded out, and then it's released into the world to spread its love and joy. Any questions on replication? Fighting, conquering viruses, which will probably end up taking us all over. Either that or fungus. Anybody watch that Last of Us show? Yeah, it's fine. That's my homie Din Djarin right there. But it's really scary because that's real. That's a real fungus. I think it can really happen to us one day, but fungus. Figured we talked about that. Won't move on. Lab diagnostics. So you're all probably sick of hearing the word gold standard by now. Hate the word gold standard. But cell culture is our gold standard. It's perfect for clinical work because it's cheap. There's a whole bunch of cell lines available. The cultures, it says that they're ideal for clinical work, but are they really? Super hard to keep alive. They get contaminated super easy, and just an overall pain. But the good thing about subcultures is it can actually help you determine between a latent and an active infection. If the viruses are actively replicating, you're going to be able to see it in the cell line. Whereas if it's just a latent infection, you're just picking it up at PCR, the culture will say, hey, this isn't really growing, this isn't really, I hate saying the word alive again because it's not alive, but it's not actively replicating, so it's obviously a latent infection. So that's really where the cell culture is good for. Good luck finding anybody to still do it for you. I guess the kids still might do it for certain things, but they were cutting it out when I was leaving there, coming over here, and we don't even send them anymore. So cell lines. I guess that since this is an intro lecture, you don't know anything about cell lines. So basically, cell lines are little vials of some sort, however they use them, and then they put a monolayer of cells. So whether they're primary cell cultures, secondary cell cultures, there's a whole bunch. And they're laid out in a very, very thin monolayer line. You put the virus in there, and it will act as if it is infecting the body. And then we can take that and we can see if it's there. We can use DFAs, direct fluorescent antibodies, to tag the cells that are infected. And we can also look for the cytopathic effect, which I think is in a couple slides here. I won't hit too much. So primary cell cultures and then secondary cell cultures. EMK, primary monkey kidney. Can anybody guess where that came from? I didn't think so. I thought that was a pretty hard one. And then your secondary cultures, these are your subculture medias. So sort of like bacteriology, but like a bacteria mycology. So like for molds and funguses, we have sap plates. That's our primary. We get it growing, and then we take it and we sub it to a potato dextrose. Sort of the same thing. We get it started, and then we can subculture it and maintain these things in the lab for a long time. You have diploid and heteroploid cell lines. Diploid are the normal number of chromosomes, and then the heteroploid are abnormal number of chromosomes. The majority of these are derived from cancer cells. So a lot of times you'll see, you know, like the MCR5 is from a fetal fibroblast from 1966. That means they have kept this cell line alive for that long. I'm not doing the math. You all can do the math, but since 1966. The HEP2 line is from a cervical cancer from 1951. So, you know, that's how we keep these things going over and over and over. They're cancer cells. They don't know when to stop. So they pretty much have a indefinite lifespan.
Pretty interesting book about that HeLa cells too. I can't remember what it's called, but it was really good. The Life of Helen. It's the lady that,
Yeah, Gila is Helen something. That's her like short version.
but it's like they kind of took those cells without
Henrietta
permission.
Lack.
Henrietta Lack. Yeah, that's
Henrietta
it.
Lack, that's her name.
It's a good book if nobody's read. Yeah.
So, yeah, continuing, continuous cell lines, these are, serially subcultured and they have an infinite lifespan. So these are your cancer cells. They're just programmed to keep going and going and going. And then you have your finites like your MCR5 that die after about 40 passes. So, didn't really tell you the benefits of each line, but other lectures are for. I just know that it's a lot of work to keep these things alive, and I would be really mad if I had to do it, it would die anyway. So, those are your cell lines. We have ELVIS cells, ELVIS, enzyme-linked viral inducible systems. These are, I want to say little cheater vials, but you take little baby hamster kidney cells, and they had a bunch of substrates, generate bacterial enzymes that are affected with HSV, and if the HSV is present, the cells will turn blue. So this is just a quick viral culture way to look for active HSV. Pretty sure this is the only thing that we ever sent to kids after a while, and then I don't know if we just stopped doing it. PCR is the way to go now for detection, but as I said, PCR, that's great and all, but you can't distinguish between an active and a latent infection. Which, if you all recall from our very first slide, that's what we're trying to do. So containers, these are our flasks that I had mentioned. They can be glass, plastic, they can be shell vials, they can be dilution plates. There's a whole bunch of different things they could be. Sources for viral cultures. So lesions, obviously, if we're gonna have HSV or shingles, what's that? I'm a brain fart. It's herpes. One of the
Yeah, herpes.
It's easy to say because they're all herpes.
Herpes.
Skin biopsies, punch biopsies, mixed tissues, urine sediments, tumors, and lymphocytes. Very interesting process getting lymphocytes for viral cultures. When I did my rotation over at kids, we did stuff with lymphocytes. It's really cool, but very complicated. So why it's so complicated? We have to keep these things alive. They're living, active, breathing cells. So like anything else that's alive, what do we need? We need nutrients. We need to be kept safe from pathogens. So we have balancing solutions. We have bovine serum, we have amino acids, pyruvate, sodium, glutamine, all those fun amino acids we need to live. And then we need to protect ourselves. Not all of those sources that we had talked about are clean, sterile. You know, you do a genital lesion for HSV and, you know, you're picking up lactobacillus, you're picking up skin flora, like negative staph, stuff like that. So we need to kill that off. So that's where the vancomycin and gentamicin come in. And, you know, same deal, fungicide, Indian albicans, normal flora in some genital tracts and respiratory tracts, just kind of everywhere. So we need to make sure that we're keeping those cell lines in peak top condition because the whole point of this survival is to look to see how the virus affects itself. We're not going to be looking at the virus itself. Can't see it unless you got an electron microscope. Who has an electron microscope in their house? I thought. I mean, I have a light microscope, but I don't have an electron microscope. So we got to keep those cell lines safe so we can look at them and see what's going on. Bacteria destroy everything. You know, you have overgrowth of bacteria on, plate, and you can't tell what's going on. You can't find your pathogen. So if you have bacteria overgrowth on your cell culture, you're not going to be able to see the cytotoxic effect, and then you're not going to be able to tell what virus. Make sense? So requirements, pretty much the same as bacteria cultures, minus the whole repeating it. Normal body temperature, 36 to 38 C, 4 to 6% CO2 ratio, the 80 to 100% humidity. Obviously, sterile technique. I mean, come on. Why do it at all if you're not going to keep it clean? Sorry, hear that when they draw blood cultures. And then we're going to constantly be replacing the medium. So I don't know, when they did the whole COVID pandemic, they showed a bunch of people with those big pipettes on the little sticks and they had things in their hand. Yeah, that was just them feeding viral cells. They like to put pictures that don't make sense. But, yeah, that's the medium for it. You're doing it, son. How long do I have?
2.
So let's do this. Okay. So collection and storage of viruses. All probably have seen a COVID-2 by now. Viral transport media. You'll see it called universal transport media that's able to hold mycoplasma, ureaplasma. M4, those are those little tiny glass vials, the little beads in it. Heck, the children used to use them. I don't know if they still make M4 anymore. I haven't seen it in a long, long time. We've all pretty much converted to a Copen UTM, but depends on where you work. So we can do either fluids directly inoculated. These would be like sterile fluids, like spinal fluid. We could do lesions. So you would take your lesion, you would swab it. You put it in your VTM. Typically, your VTM has vancomycin and the same stuff in it that you're putting in it, so it kills off the bacteria. Stool, unfortunately, if you're still doing stool cultures, stool viral cultures that way, you're going to put a lot more work into it because there's a lot more bacteria. So you dilute it out into the VTM, which would hopefully kill off most of that, and then you can inoculate it. Store transport media at 4 degrees until planted, refrigerated. And then viruses, we can cryogenically freeze at -70. So we can pretty much indefinitely store viruses. So we have our -70 stock full of a bunch of viruses. During COVID, we had still got to stop talking about COVID. Detection of viruses. So, as we said, gold standard, we'll do cell cultures, and we're looking for the cytopathic effect. I think I've mentioned that a couple times. Shell vials. These are centrifuge of the specimens into a cell sheet. So basically, they're almost like a half a test tube, but at the bottom of the test tube is flat and it has a cell line in it. Put your sample in. You centrifuge it down and basically the virus into the cell. It just kind of helps speed up the process a little bit. Immunofluorescence. There's a label on the antibody, and it goes and then finds the antigen it's looking for, and then you can look at it with a fluorescent microscope. DFA. Direct fluorescent antibodies. We use that for rapid detection of HSV, influenza. EIAs, those are your little quick strips. You mix it up and then crosses over the barrier and yada, yada, yada. And then PCR, which is what 90% of labs do nowadays. While it is very helpful, it's still not helpful to distinguish between it. Like, I've put that a couple of times. So this is what our PCR respiratory panel looks like. This is the BioFire 2.1. So this is the main virus multiplex PCR that we use. So we can see here on our little picture, adenovirus, or coronaviruses, metapneumo, a little blip about rhinovirus, enterovirus. You can't genetically distinguish them, so we'll cover that a little bit later. Influenza, parainfluenza, RSV, and then it throws some bacteria on it. All right, clinical significant viruses. So these are the ones that we're going to see on most or on a more daily basis. Things that we're all probably going to have at one point or another. So our common respiratory viruses, adenovirus, coronaviruses, which I loved explaining to people. You know, they see they have a coronavirus, and like I said, is that a problem? Yada, yada, yada. And like, no, it's not. It's just a cold. And then here we go. Everybody coming saying, oh, I thought you said it was just a cold. Well, it wasn't. So we have hundreds of coronaviruses. Most of them are just a standard cold. Everything in the top row, and then you have your severe acute respiratories and your MERS, Middle Eastern respiratory. Rhinovirus, that's your typical everyday cold as well. Metapneumovirus, that a lot in the elderly. Influenza A and B, obviously you all know what that is. There was something going around saying that during that whole COVID pandemic, when everybody was masking up and stuff, that we may have actually eradicated influenza B. You ever hear that?
Mm-mm.
So there's only 2 strains of B. We'll get to that when we cover influenza, but there was between the 2 or 3 year time span, there was not one detected with the B. So they think they might have eradicated that. We'll see for that.
That's nice.
A still around. Para-influenza 1, 2, 3, and 4, and then RSV. RSV is the one that takes out the little types. Are they doing something or somebody?
I was told that. I know, I think it's weird, so I'm gonna turn it off when he's in the kitchen and he's scared because the heat is taking.
It calls the student way.
Yeah. One, those are. Like releasing.
We're all gonna die.
That's the worst thing that happens to you today.
I'm glad it's my thing. Two decades.
So, anywho, common enteric viruses, adenovirus, astrovirus, norovirus, and rotaviruses are 2 big ones. And then saproviruses, 1, 2, 3, and 4. Skin and tissue virus, that's your Coxsackie, that's your hand, foot, and mouth. Herpes, obviously, we all know what herpes is. I'll probably jam for that too, never mind. HPV, and then BZB, your herpes zoster, varicella zoster. Pretty sure everybody has had contact with one of these, maybe once in life, if not multiple. CNS viruses, these are your, viral meningitis, encephalitis, stuff like that. CMV, problem with a lot of babies. Enteroviruses, your poliovirus, hopefully that doesn't come back anytime soon. But you have your Coxsackies and your virus. Herpes, HHV6, human herpes virus 6. We get a lot of that in spinal fluid. Just had one not too long ago. Paraequivirus, So Foster, and then West Nile. I remember when West Nile came into that was fun. I was freaking out about it, but it was only pregnant, so I was safe there. So what I did for these next, we're kind of gonna break them down now, but I put what kind of virus they are, so that kind of helps you figure this out. So positive sense, single strand RNA virus, we talked about it, those are the ones that can just go right in there. So coronaviruses, we saw how, everybody is finally brought to light about coronaviruses, so this kind of helps tie those two together, I guess. Our four common strains, these are mild upper respiratory tract viruses, your COVID-22-9E, your HKU, your NL63, and then your OC43. They all have some significance, I guess, to wherever they're found. I think OC is more likely, but it's just me guessing. But they're all just your common colds. You get them all the time. People had no idea what that meant. So I would just tell them it was common cold. And then here comes SARS. SARS was just a little bit before my time, before I started working in a lab. But that's your severe acute respiratory syndrome. Obviously, caused a very acute respiratory problem that was very severe. So it was severe. Then came the Middle East. Respiratory syndrome, and then they're out of order, but we just obviously had the second round of coronaviruses that cause severe acute respiratory syndrome. So, SARS, transmission through respiratory droplets and fomites. Basically, you get high fever, flu-like symptoms. Diarrhea was going around for a little bit. Hypoxia, obviously, you're having lung damage from the release of the organism, the virus, and you're going to have a hard time breathing. And then most people develop pneumonia, inflammation from that. A lot of the times you develop secondary pneumonia. We were starting to see a lot of cases of people that were coming down from SARS. We would see an increase in weird atypical like pneumonias, things that we would consider normal flora, but really weren't. Like candida pneumonia and stuff like that. So, anywho, how we identify PCR serology. When this first came out, and especially when the second one came out, it was a Class A bioterrorism organism. If you were to culture it out, cell line. If you did PCR, it wasn't. It was B, Class B. COVID-19, I haven't really updated this slide since it originally came out, but it stands for SARS-CoV-2. No longer novel. The ACE2 receptor, that's what it was. I knew it was something like that. So you're talking about how it has affinity for those receptors. And what was really, really, really super weird, not going to get into the conspiracy theory aspect of this, purely science, it was an exact match to the human ACE2 receptor, which is weird because coronaviruses are bat viruses. Yeah. So anywho, some magical way, maybe not, change of function passage through a pangolin. They have adapted the ability to bind to our human ACE2 receptors and not come up. So that's what made it so bad. Really, upside, if you want to say it's an upside, we came out with these very new and very exciting, even though they're not new, but they're new in people. mRNA vaccines. So you all have heard about it. You all probably had to have gotten one, probably. We had to. So basically, this is a technology that has been there for a while, but it just kind of played a role in veterinary medicine, vets, stuff like that, animals. But then they're like, well, if we can do it in a dog, why can't we do it in a person? And it seemed to work. So if we can do it for this and make a vaccine like that, we can cure AIDS, we can cure, herpes, we can cure CMV, we can cure anything. So, there's a lot of this and that, and, you know, the government's putting stuff in the body and gonna change the need, it's not, look into it, do your own research. I'm not here on a soapbox about those, but really cool. Human herpes viruses, these are going to be double-stranded DNA viruses. So they take time to replicate. They're not something that's, you know, hits you like a cold real quick and then just kind of goes away. Most of these are latent, they stay with you forever. So they're slower, they don't have as much, you know, hurrah when it comes to releasing, causing a lot of cell damage, you know, viruses, they want to stay active as long as possible, so they don't necessarily want to kill the host right away. So, classification of herpes viruses, you'll see HHV as the designation. So you have 1 through 8. Your HHV1 is your herpes simplex 1. HHV 2. HHV 3 is your BCV, your varicella. 4 is your Epstein-Barr virus, trichomononucleosis virus. 5 is CMV, cytomegalo. HHV 6 is the Roseoleo virus. We just call it HHV 6. 7 is pretty much the same. You see 6, 1 to 7, 79. And then 8 is your Kaposi sarcoma. We typically will see that associated with all those HIV/AIDS patients. Anybody watch American Horror Story? That last one, the NYC,
Yeah.
really good because you thought this was a serial killer and it was about HIV. It was really cool. And then he dies of that. So.
Check it out.
All right, where we at? So, HSV, we all probably have heard of it at least one point in time. We can find this in respiratory, in direct contact. So, you know, HSV pneumonia, meningitis, cold sores. We usually see HSV1 with that. And then your HSV2, that would be your sexual contact. That would be like your genital herpes. Fortunately, you can have either one in either place. So it's kind of hard to tell. That's why we differentiate them. Not that the treatment is any different. You don't treat it. You can't treat it. You treat the symptoms. So you got it for life. So signs and symptoms, cold sores, genital lesions, viral meningitis. Epidemiology, you know, HSV1 is your face or can be genitals, but it's not typically that. We typically see 2 as the genital. And then neonatal infections, obviously, those are acquired during birth as they pass through the infected area. You need Aclavir and Valcalavir to treat these. Usually it's the Aciclovir. I'm not a big antiviral person. Cyclovir. That's an exciting one. We see that a lot in Dr. T's reports. So basically, you can use antivirals to slow it down. Wait till it gets back into that latent phase. But there's no vaccine available yet. But you know, who knows? We've got mRNA vaccines now. How we tell PCR, our number one way. You can do serology. You can do shell vials, and then obviously shell culture. But they take a lot longer. Somebody has HSV meningitis, you want to know if it's possible, get them on antiretrovirals or just antiviral, not retrovirals. VZV, these are your chickenpox. Direct contact respiratory, that's where you had those little chickenpox parties back in the day. Pretty sure those are frowned upon nowadays. Infectious 1 to 2 days before and then 3 to 4 days after. So that's why it's kind of really bad because you don't know when it's going to start because you're infectious before you even get symptoms of it. And then, long after they erupt, where most things, once they erupt, and, they dry up and scab over, you're done. So you're itchy, you got a fever, got nice little red pox everywhere. Tended to be seasonal in children for chickenpox, and then unfortunately for wonderful adult community that's had chickenpox, that can be reactivated and cause what I hear is awful as shingles. Therapy is the same, but we do now have a vaccine available. So that's why they'd rather have you do a vaccine than get the chickenpox or pox party, but anywho, vaccines.
Are you old enough to go to a pox party?
I was,
Yeah,
I was
I
a
did too.
pox- I was a pox party baby.
Me too.
I turned out just fine. Doing your lab ID, obviously PCR, virology, shell bios, shell cultures, they're all pretty much the same. I think we are now working on building an in-house research here. Got a lot of serology for that, but. Epstein-Barr, love the name of that. So many ways to use it in jokes. Anybody watch The Family Guy? I think they did an Epstein-Barr joke where they all go to Epstein's bar. It's anywho. You're all probably familiar with mononucleosis. So the kissing disease, you get it from saliva, oral sexual contact. It takes about 2 to 3 weeks and it stays with you for life. You get hepatosplenomegaly, you get that nice big swollen spleen, then hepatic fatigue, sore throat. And then you go and find that person you kissed and be like, what did you do to them? Unfortunately, that can also cause Burkitt lymphoma, nasopharyngeal carcinomas, and other cancers because they contain parts of the EBV virus. So there's been a lot of theories and a lot of speculation. I didn't really look into it too much. But people that are infected with the HBV virus, they're the ones that are starting to have a lot of these issues, like gluten allergy, that. Stuff like that, like cancers and all these other autoimmune disorders, because they're thinking that they had eventually been infected with Epstein-Barr and then it's caused mutations. So, I don't know, it could be true. I can tell you I did not have a gluten allergy when I was a kid. Now, if I touch the stuff, I break down and it's rash. So,
Did you have mono?
I did. So, who knows? Lab ID, this is the one that we don't really culture out. I don't know why. Serology is how we typically do this. We'll throw a little bit of blood, we'll throw it in the tube, stick a little quick strip in there, and then we can tell if you had mono. Super easy test. PCR we reserve for spinal fluid. Obviously, if they're drawing spinal fluid, it's serious enough. So you know it's on the spine, so pay a little bit more money. You got human CMV. Oh, we're running out of time, so we're going to go through these pretty quick. CMV babies, babies get that a lot. You get fevers, jaundice in infants. Only humans are known to carry this, and it's concentrated in the salivary glands. Therapy, no therapy. Basically, most of these are just support care. And then PCR, serology, urine. Urine's another one we can do. Enterovirus, these are your Coxsackie, Echo, Rhino, polio viruses. Coxsackie, your hands, foot, and mouth. See that a lot in like the football team or toddlers, infants. I know that big went around when I was playing high school football, but I didn't get it because I didn't touch those during water loss. That's probably why I'm a microbiologist. But epidemiology, it's typically summer, autumn football season. No real therapy prophylactic. She gets supportive care. They'll give you some, steroid, help with the rash. PCR, shell vial, one of our. Echovirus is your enteric cytopathic human orphan virus. Echo. No, not Alexa, but I had to change it to Echo because every time Alexa was on TV, it would go off, so now I have to say Echo. Anywho, fecal oral, obviously enteric. Cough, rash, flu-like symptoms, rare viral meningitis. I may have seen it one time. I've been here. Therapy, supportive care, PCR. Rhinoviruses, you've all have had a rhinovirus. If you haven't had a rhinovirus, you're not a human. So, respiratory transmissions, non-productive cough, headaches, general malaise, and feeling like crap. Varies for the seasons, typically this time of year. Most frequent infections in the man, and in general, even though my dog sneezes a lot, so I think it's cold. Known as the common cold, and there's over 100 serotypes, so can't really make a vaccine for it. Who knows? One day. Supportive care, PCR, shell culture. Polio viruses, fecal oral, general headache, malaise. Poliomyelitis eventually will cause cranial nerve damage. That's why you got the people in the wheelchairs. Some of my older coworkers that are no longer here, when they got the polio vaccine, it actually caused some paralysis in them. So they walked with a limp because the original polio vaccine used polio. So it caused partial paralysis in a lot of these people. Hers wasn't terrible, you didn't know unless she told you, but then when you actually look at it, you can see the limp. PCR, serology, shell vials. Hopefully you will never see this in your career unless you work for one of the military labs. Adenovirus we can get pretty much anywhere. Respiratory, fecal oral, direct contact. Respiratory tract infections, GI infections, conjunctivitis. No cure. We can do EAAs and DFAs for these as well. They're just so abundant. These are the strains that we can have for different for different sites. So respiratory conjunctivitis, gastritis, and they have tried to associate adenoviruses with obesity. Influenza. These are the ones that we really all love and know how much got my flu shot yesterday in the conference. Plus the flu shot. I don't know how it's treated, but I'm not going to cover that. You all know what it does. We're not going to cover that. You all know when it's coming. What you need to know about the flu is the surface antigens, your hemagglutinin and your neuraminidase. That's your H1, your N1, your H2, your N2. That's how we classify based off of the surface proteins. I have a crap ton of antivirals. Anything that entered the beer is a viral, antiviral. They have new ones now that are probably way past this. And then we have our seasonal vaccine you'll come to know and love. They have live, they have attenuated, they have nose spray. They come in all shapes and sizes. PCRs, we do it now here, but you can do DFA, you can do EIA, and you can do cell cultures. So you got A, B, C, and D. A and B are human ones. C and D are everything else. Don't even think we touch or C. So the big takeaways, because we're running out of time here. Hemagglutinin and neuraminidase. That's how we classify it. H1N1. Antigenic drift versus antigenic shift. Drifting is just like a small, you just kind of drift out of your lane for a little bit. And antigenic shift is when you shift into a completely other lane, you go full body into it. So that's your major changes. Here's a couple of fun strains for you all to look over. But notice how many strains are on this page. Now, B, we have two. We have the Victoria and the Yamagata strain. They're not further divided into subtypes. This is, like I said, there's only two of these, and I think they were completely wiped out. But I'll have to do more research into that because took over. How we name them, it's important to know this part. It'll probably be a question somewhere in life. Your virus type is first, location, strain, Viro is isolated, and then any viral subtypes. So you can read over that. Paraflu, it's been reclassified like everything else in this world. But most people still know it as parainfluenza, so most people keep it as parainfluenza. Croup, if you're anybody familiar with croup, have small children, you know what croup is. That's your type 1 and type 2 parainfluenza, which are now reclassified as human respirovirus 1 or human rubeolavirus 2. Why? I don't know why they had to do it. But anywho, they reclassify everything. We know it as parainfluenza. We're going to teach it as parainfluenza. Just be aware that they change the name. So 1 and 2 are the groups of the respiratory tracts. Then you can have the other two that just are plain, boring, run-of-the-mill respiratory illnesses. Same thing, flu, coronavirus, it's all the same. No treatment, no cure, just, supportive care. Summer variations, or seasonal variations for spring and summer months. RSV, this is another one of the big ones. Droplets can cause upper and lower respiratory symptoms, typically in the winter. So we're getting right into that good old HSV, RSV influenza season. Ribavirin produces viral shedding. I have heard rumors of a new vaccine for RSV. Not sure when that's going to be approved, if that's going to be approved. But PCR, EAA, DFA. Kids used that Sophia for a while. I don't know what they do now. They might use the Cepheid now. Measles, mumps, rubella, MMR. Should all know that there is a vaccine for that by now. Self-limiting, so there's no therapies. Spread by respiratory droplets, very contagious. And then these Kepler spots, which are raised red spots about molars. Measles, bueno. You would think all this stuff would be eradicated by now, but don't get me started on anti-vax. Anybody anti-vax here? If you are, I'm going to make fun of you, so just keep it to yourselves. These were all pretty much eradicated, and then you got these people that think the government's trying to, whatever. Live your life, I guess. Just don't bring it near my child. Transmission of mumps. Anybody ever watch what is that, Brooklyn Nine-Nine, where they get the mumps and they got the big. Yeah. So another respiratory thing. Painful, swollen salivary glands. That's why their necks were really big. Can turn into meningoencephalitis. Not good, no bueno. But if you are a post-pubertal male, you get swelling of the testicles, which I don't imagine what that feels like. Sorry, guy, we're the only ones here, maybe in the middle, so would not be fun. Like most viruses, self-limiting, it's part of the MMR. So you all should hopefully be vaccinated. And then serology is how we look for this. Cell culture. Rubella, same kind of deal. Should really be gone, but it's not. We get a lot of this in congenital. You'll have a lot of what's the word? Prenatal screening for this. Because, in the first month of pregnancy, you have a 15% chance of spontaneous abortions. First month of infection, you got a 100% chance. So, it's not good. Seasonal, summer, no therapy, and MMR. Rotavirus, it just makes you poop a lot. You can do PCR, EAA, a little cassette test for these. Winter, spring, prominent children. There's a picture of it. Asperoneurosapro, this just kind of covers the rest of the fecal viruses. Asperoneurosapro, they're okay, cause a little discomfort. Norovirus, my wife had it once, it was nasty. Norovirus sucks. There was a big outbreak of it not too long ago. All three of these are on our PCR panel, so it's all encompassed into one PCR test. But in areas that have a high prevalence, they might have EIAs for quick screening. Doctors' offices don't get them. HIV, we're just going to hit real quick because you got a whole lecture on it. Below, I swear it was a double screening virus. I'm not an HIV expert. Dr. England is. So HIV obviously is what causes AIDS, acquired immunodeficiency syndrome. It is a retrovirus, worldwide, not seasonal. They have highly active antiretroviral therapies. Usually they'll give stuff like that to babies, stuff that come out of an HIV positive mother, or with your exposure protocol, like in a hospital when you get stuck, they'll shoot you up with a bunch of these antiretroviruses, hoping to stop it before it can take hold. Lab identification, EIA. Typically, this is like your rapid EIA. And then once that's positive, you send it out for Western blot. Western blot's always been the gold standard. For confirmation of a positive rapid, you can isolate the virus. I don't know why you would, but you can. And then you can do PCR. I don't know why not on here, but we test for the P24 antibody. Here at SUMA and probably in Altman and everywhere else in the world, because that's the new standard. So you just draw a blood tube, we look for the P24, and if it's positive, we will then reflex it to confirmation by PCR, do a viral load. I don't know, do you guys send out for Western blot?
Yes. Think they do.
We used to.
Does Altman?
I think the doctor has to request it, but we.
Yeah,
we
if
do.
they request it, but not just automatically.
Not automatically, I figured all,
Yeah.
because when we do the PCR, we can quantitate and do a viral load off of it, so like cheaper.
Right.
HIV retrovirus, like I said, we're out of time. You're gonna have a whole lecture on this. This is really just an intro, so I like to get into too much stuff I can for. So, one of the big takeaways with these is they do the reverse transcriptase. They transcribe viral RNA to complementary single-stranded DNA, and then they integrate in the cell host. And it causes AIDS. You get 4 symptoms: infectious, asymptomatic, symptomatic progressions from HIV to AIDS, which all relies on your CD4 count. Do flow cytometry will get your CD4 count. Falls below 200 cells, you have AIDS. Make sense? Cool. Human T cell leukemia virus, HTLV. It's very similar. It's a retrovirus that affects the T cells, spread through blood, sex. So it's literally like a little cousin of HIV. No cure, no treatment. It's all the same stuff. Hepatitis, you have A, B, C, D, and E. A is not too bad. B is the worst. C is curable now. D I've never heard of, and E, cool. As the name foretells, hepatitis affects your liver. I've had it. It's not fun, but it's not the end of the world. Like I said, B is curable, not curable. B is not curable. C is curable now. So A, contaminated food and water, it's the chi-chis. If you don't know what I'm talking about, look it up. There is a vaccine for this, so, get vaccinated or get a chi-chis. Hep B, that's the big one that everybody worries about. It's non-curable. You can treat it, symptom wise to keep your liver from dying. But this is the one that, they check for when you go to work. They give you the vaccine. And you get all those different tests, which are described right here. You got your surface antibody, your surface antigen, your core antibody, and your IgM. So there is an algorithm for all this, and once you figure it out, it's super easy to understand, but we don't have time to explain
I
it.
have another lecture on this too, so.
Perfect. So this is your interpretation chart, cool. Hep C, here for was discovered in 13, it's starting to be more prevalent. Hep, it's Delta, it's not D, by the way. Like I said, never heard of it until I started this program and stuff. But then E, that's your shellfish and stuff like that. HPV, everybody has HPV. Don't care what you say, you have it. Just might not be the bad one. There's 10 out of the 30 HPVs that are considered cancerous or associated with cancer. So that was all your other ones. Arboviruses, these are your ones that are all associated with arthropods. Like I said, if it comes from an arthropod, you're screwed. Togavirus, Filarovirus, Viridae, and Bunya viridae, those are all your classifications, your chickpea, your triple E, and your wheat. Those are all your equines, and chicken do I guess chickpea, call it chickpea, watch it all, you'll figure it out. Those are the pretty nasty ones. We do have triple E up in Michigan and Boston and up there. There was an outbreak not too long ago. On the East Coast, obviously it's called Eastern equine for a reason. Western, I'm assuming, is on the West Coast. I don't live over there, nor do I desire to. Then we got our Filaroviiridae. That's your dengue fever, your West Nile, your yellow, your Zika. And then your lacrosse, your LAC. Arboviruses, arthroborne, transmission through blood, triple E, your WE, your SLE, and your LAC. Those are your big four. PCR, we do a lot of PCR and spinal fluid and serology for everything else. West Nile, mosquitoes, transfusions, and pregnant mothers. Read up about all that. Reportables, you have your class A, anything class A is bad. That's your fevers, anything with fevers. Marburg, Western virus, Ebola, monkeypox, smallpox. Nobody knew what monkeypox was until recently, which doesn't really seem as bad as it looks, but I haven't had it, so I'm not going to judge. Rabies viruses, you hear a lot about rabies. There's really nothing you can do for that. You die of craziness, and then the only way to confirm it is by cutting your brain out. Those are caused by Lassa viruses, and then COVID was a class A for a little bit. Any questions? Sorry, we got real quick there, but I finished it too. Yes.
Rabies, isn't there like a time period where you have it's able to be cured? Or if you get big, you're automatically screwed?
From what I understand, it's sort of like the HIV and the exposure protocol. There's a certain time frame where you can get those shots in your stomach, and you can prevent it from being able to penetrate your cell and start to replicate. And I don't know for 100%. Somebody.
That's true. I used to work in veterinary medicine, and that was the protocol. You had to put the animal into quarantine for 10 to 14 days to figure out if they actually had the rabies virus, even they had the vaccine. And then you had the choice to go and it's a 3-shot series for rabies to treat yourself.
There you go. Yes, sort of like the HIV, exposure protocol. You try to stop it. So obviously, knowing when the infection occurs or when the potential infection occurs is important.
One question.
Sure.
With the herpes simplex virus, you stated how you can have either
Mhm.
in any location of the body. Does it depend on where you are infected, what you have, and if it can be transferred anywhere?
You have oral herpes, it can be deposited on the genitals.
Okay.
I don't want to explain why, but And vice versa, if you have genital herpes and it ends up in your mouth somehow.
It's probably not just gonna like magically sprout up.
Yeah.
You have
Well,
to come
see,
in
the cool thing
contact.
about- The cool thing
probably.
about herpes though, is herpes virus, it travels along your neurons. So, it starts out down there and it kind of goes away, when you have these flare-ups. Well, it likes to travel along those nerves, so it's just kind of like a roller coaster inside your body until it wants to come out.
So it can.
So, anyway,
And I think that's how the shingles,
and that's how.
why the shingles come up everywhere, but
And because the herpes viruses are all the same, it's sort of the same. So it could, start down there and come up here.
and also I remember hearing about it causing like something severe in the brain, like
You can have pterygium encephalitis.
Yeah.
From viruses, especially HSV, because like I said, it travels along neurons. So, we do get HSV quite a lot.
yeah.
in Environment and things. We get a lot more viral than we get bacterial. And most of the time, it's either HSV, VZV, or homa. I want to say HHV6. So it's one of the herpes viruses. So because the herpes travel, cases.